we have learned through the various types of bacteria and also viruses. So now in this part, we're going to learn the antimicrobial agent, which means the drugs or the medication that can be used to inhibit or kill pathogen. However, basically here what we're going to focus is only the antibacterial. This is because, as I told you previously, that currently actually we are lacking of this antiviral for many viruses. So the main focus here is only focus on this antibacterial, which is the antibiotic. However, the term antimicrobial is applying for those drugs that against bacteria and viruses. So first of all, we come to a few of the definition. First, the definition antibiotic. So this antibiotic generally is referring to those drugs that used to inhibit growth of bacteria. While for antimicrobial is any substance with sufficient antimicrobial activity that can be used in the treatment of infectious diseases. This is referring to bacteria and viruses. While for bactericidal and bacteriostatic, what is the meaning? So bactericidal is referring to an antimicrobial that not only inhibit growth but it is lethal to bacteria, which means bactericidal is the antibiotic that can kill bacteria. While bacteriostatic, static mean no change, which means no increase in growth. So bacteriostatic is referring to those antibiotics that inhibit the growth of bacteria, but it doesn't kill the bacteria. So this is the difference between bactericidal and bacteriostatic. Bactericidal will kill the bacteria, but then bacteriostatic, it doesn't kill the bacteria. It just inhibit the bacteria from continue growing. So these are the example of antibiotic that have this bacteriostatic property, while these are the example of antibiotic that is showing bactericidal property. However, one thing you should bear in mind that it doesn't mean that, for example, erythromycin. So erythromycin is referring to bacteriostatic, which means it does not kill the bacteria, but it just inhibit the growth of bacteria. But it doesn't mean that this erythromycin is forever belong to bacteriostatic. You have to bear in mind that it is also affected by the concentration used. Sometimes, if you use excessive amount, very high concentration of erythromycin, it can also become bactericidal, which means it will kill the bacteria also. So bear in mind that this is affected by the concentration used as well. Very high concentration of antibiotic which belong to bacteriostatic may cause bactericidal effect as well. Minimal inhibitory concentration. This is a laboratory term that referring to the lowest concentration that able to inhibit growth of microorganism. What is the significance of this minimal inhibitory concentration, or we call it as MIC? This is important so that we can know what is the minimal concentration of antibiotic that should be used to kill the bacteria, so that to prevent toxicity in the host. Example like toxicity in human. And some bacteria they are unable to be inhibited by the specific concentration of antibiotic. So we say this bacteria is showing resistant to the antibiotic. Some bacteria are sensitive, which means they are able to kill by the particular antibiotic with the particular concentration, the MIC. Spectrum is an expression of the categories of microorganisms that against which an antimicrobial is typically active. Some of the antibiotics they have broad spectrum. Some of the antibiotics they are narrow spectrum. This one we'll discuss later. Susceptible is a term applied to microorganisms indicating that they will be inhibited by concentration of the antimicrobic that can be achieved clinically. Simply, susceptible is meaning sensitive. So a bacteria which is susceptible to antibiotic, which means the bacteria is sensitive to the particular antibiotic. So now, first of all, where do we discover all these antimicrobial? Where do we discover all these antibiotics? So typically there are three main sources. The first is from fungi. So normally these antibiotic they can discover for fungi. Example like penicillium. Penicillium antibiotic. Penicillium is a fungi that produce antibiotic penicillin. While the second source is from Streptomyces. Streptomyces is the type of soy bacteria. This soy bacteria produce a lot of antibiotic that can be used in clinicals nowadays. So these are the example of antibiotic that produced from soy bacteria, Streptomyces. The third is the largest group that normally we produce through this molecular manipulation of previously discovered antibiotic, which means you already have one antibiotic. We do some molecular and chemical manipulation to produce a different type of antibiotic. These are the third group of antibiotic. So in summary, there are three sources of antibiotic. First is from fungi, second is from the soy bacteria, Streptomyces, and third is the molecular manipulation of antibiotic. Just now we did talk about spectrum. Antibiotic can be grouped into narrow spectrum and broad spectrum. Narrow spectrum, which means the antibiotic that against only certain specific group of bacteria. Broad spectrum antibiotic is the antibiotic generally can against many type larger group of bacteria. So in clinical, we prefer which type of antibiotic? Generally, if we know that the type of infection, we will go for narrow spectrum, which means if you can know that this infection is caused by which bacteria, we always go for narrow spectrum. However, if the cause of infection is unclear, then we will go for broad spectrum. One of the example is amoxicillin. So amoxicillin is one of the commonly used antibiotic that being used when the source of infection is unclear. Next, we're going to look at how the mechanism of action of this antibiotic. How can antibiotic inhibit or kill bacteria? So we can summarize them into four. First, the antibiotic can kill or prevent the growth of bacteria by inhibit the cell wall synthesis. Some of the antibiotic act by inhibit the formation of bacteria cell membrane. Some of the antibiotic act by inhibit the protein synthesis in the bacteria, while some of the antibiotic interfere the the DNA replication, or we say the transcription of this DNA or RNA in the bacteria. Let's look at the first one. So the first category of antibiotic, they act by inhibit the cell wall synthesis. So normally this is belong to those antibiotic that belong to the beta-lactam group and also glycopeptide. Beta-lactam group is those antibiotic having this beta-lactam ring. So example like penicillin, cephalosporin, and also carbapenem. So the picture here showing the beta-lactam ring. These all are the beta-lactam antibiotic. They're showing this beta-lactam ring. This is the beta-lactam ring. This is the beta-lactam ring here and this one. So those antibiotics that having the beta-lactam ring, they belong into this group. So they inhibit the bacteria by inhibiting the formation of cell wall. So what is a component of bacteria cell wall? Bacteria cell wall, one of the main component is peptidoglycan. So the mechanism action is very simple. This antibiotic can inhibit this formation of peptidoglycan either by inhibiting the enzyme that synthesize this cell wall. So you can see in this picture, this is the bacteria cell wall. So the bacteria cell wall formed by the amino acid residue. So in normal condition, in a normal scenario, the bacteria will need a type of enzyme. This enzyme is called transpeptidase enzyme. This enzyme transpeptidase is important to be used by the bacteria to synthesize their cell wall. This enzyme transpeptidase, also known as penicillin binding protein, short form we call PBP. So what is happening is, for example, like antibiotic penicillin. Penicillin will bind to the transpeptidase enzyme. Once this antibiotic penicillin bind to the transpeptidase, by this the enzyme is unable to catalyze the formation of cell wall. So without the formation of cell wall, this bacteria will die. So that's why this enzyme transpeptidase, also known as penicillin binding protein. However, some of the beta-lactam antibiotic, example like vancomycin, this vancomycin, it doesn't bind directly to the enzyme transpeptidase. Vancomycin bind directly to the amino acid residue. This makes the enzyme transpeptidase unable to bind to the amino acid residue. By this, the enzyme cannot catalyze the formation of bacterial cell wall. So, in summary, these type of antibiotic, they can act by directly inhibit the enzyme which synthesize bacterial cell wall, or they bind to the amino acid residue directly to make the enzyme unable to catalyze the linking of this amino acid residue. The second group of antibiotic act by inhibit the formation of bacterial cell wall. So example like the antibiotic that belonging to polypeptide group, example like polymyxin B and colistin. So the role is just like the detergent, which means because the cell wall is formed by phospholipid, so they act like detergent to rupture the phospholipid. So when the cell membrane is ruptured, the cytoplasmic content will be leaking. Losing of the cytoplasmic component will make the bacteria die. The third group of antibiotic inhibit or kill bacteria via inhibiting the protein synthesis, mainly translation. So this antibiotic example like is belong to the group of aminoglycoside. Example like gentamicin, streptomycin, tetracycline, chloramphenicol, and microglyc. Example like erythromycin. How they act? For example, you can see in this picture, this is a process of translation. This is a mRNA, this is a ribosome, and this is a tRNA. So example like the one that labeling A, this is a aminoglycoside group antibiotic. These are antibiotic, they prevent the formation of this initiation complex, which means they inhibit this tRNA to bind to this ribosome, so that to initiate the translation. So by this, without translation, without protein. So without protein, then the bacteria will die. And then the other one, example like the one labeling T, which is tetracycline. So they act by the same manner. So they bind directly to the 30S ribosome to prevent translation. While for the one labeling C, which is chloramphenicol, this one it inhibit formation of peptide bond. Without peptide bond, the amino acid is unable to be linked together. Without the linking of amino acid, polypeptide is unable to form. While the other one, example like the one labeling E, which is erythromycin. This erythromycin prevent the movement of tRNA to the next position in the ribosome. By this, inhibit elongation of polypeptide. Well, the question you might be curious, why? Without translation, the bacteria die. Without translation, which means you will have lacking of formation of polypeptide. Without polypeptide, which means the bacteria will be lacking of essential protein or enzyme that for their survival. Example, the bacteria will be lacking of enzyme, which is very important for respiration. Without the ability to undergo respiration, then the bacteria cannot survive. Then the bacteria will die. The fourth group of antibiotic that inhibit bacteria is they inhibit the nucleic acid synthesis. This is referring to inhibiting the DNA replication or transcription. So example like the bacteria, the antibiotic belonging to the group of quinolone, example like ciprofloxacin, one of the commonly used antibiotic in clinical, folic inhibitor like sulfonamide, rifampin. So you can see example here. Okay, example for sulfonamide. This sulfonamide inhibit the protein synthesis, so they inhibit the DNA replication. While for antibiotic like rifampin, they inhibit this RNA polymerase enzyme by this inhibiting transcription, inhibiting the mRNA synthesis. For example, the other example like this antibiotic quinolone. Quinolone will inhibit DNA topoisomerase enzyme. This topoisomerase is important for DNA replication in bacteria. So by inhibiting topoisomerase, the bacteria is unable to duplicate their DNA. So in summary, we can say that this antibiotic act on four ways: either inhibit the cell wall synthesis, inhibit the nucleic acid, which is DNA or mRNA synthesis, inhibit the protein synthesis, while the other one is inhibiting the formation of cell membrane. You may be curious what are the side effects of this antimicrobial agent. So sometimes it may be leading to toxicity. That's why the toxicity study is very important to identify what is the minimal inhibitory concentration that should be used. When some patients, they may be allergic to this antibiotic. Example, like those patients with G6PD deficiency. Sometimes the frequently use of antibiotic may also rupture the normal flora. By removing the normal flora, it increases the chances for those pathogenic bacteria to occupy the area which previously occupied by the normal flora. And what the factor may influence the effectiveness of antibiotic. So sometimes the drug ability to reach the site of infection greatly influenced by the mode of transmission, either given by oral, topical, or this parenteral, and sometimes also different, and also the different area. And also the susceptibility of pathogen, as some of the bacteria, they develop this antibiotic resistance, and also is influenced by the growth rate and also by the inherent properties such as whether there is no target for the particular drug. And also is the drug concentration in the body. So the drug concentration of antibiotic use, it must exceed the pathogen's MIC. And as we said, some of the pathogens, they develop drug resistance. This makes the antibiotic cannot be used. So in this scenario, we have to use different antibiotic, or we need to use combine two type of antibiotic. So it's very important to determine the level, the MIC. So how do we determine the MIC of antibiotic? It's very easy. You can prepare a series of broth, nutrient broth, and then in the nutrient broth is added with serially diluted antibiotic. So from the serially diluted antibiotic, we're going to looking for the lowest concentration at which there is no bacterial growth. So the lowest concentration of the antibiotic that causes no bacterial growth, that is known as MIC. So now what is happening is, now take all those test tubes that there is no bacterial growth. Okay, not only the MIC, just take all the test tube that's showing no bacterial growth, grow them in the fresh medium, and this fresh medium do not have any antibiotic at all, looking for any new growth or not. So from there we can know what is the proper concentration that should be used to inhibit the growth of bacteria.